Have you ever wondered what happens inside your cells when you don't eat for several hours? Well, it turns out something remarkable begins to happen. When the body goes without food for a longer period of time, around 16 hours, cells start to activate an internal cleaning system called as the autophagy. Autophagy, it literally means self-eating. But it's not harmful, instead it's a highly organized recycling process where the cells break down damaged parts and they reuse the materials. In this process, the worn out proteins, broken organelles, and cellular debris, they are captured and they are delivered to structures called as lysozymes, where they are degraded and they are recycled. The result is a cleaner, more efficient cell which can repair itself and maintain energy balance. This cellular cleanup system was so important that it led to the 2016 Nobel Prize in Physiology of Medicine awarded to Japanese scientist Yoshinori Ohsumi for discovering how the autophagy works. To understand autophagy, imagine a cell that has become cultured with damaged components. Proteins can become misfolded, mitochondria can wear out, and harmful pathogens can enter the cell. Autophagy acts like a biological recycling system. First, a small membrane structure starts to form around the damaged material. This structure eventually it closes into a vesicle called as the autophagosome. The autophagosome then fuses with lysozyme, which contains powerful digestive enzymes inside these compartments. The damaged components then they are broken down into basic building blocks like amino acids, fatty acids, and sugars. These molecules are then reused by the cell for energy production and repair. Because of the ability to recycle materials, autophagy helps cells to survive stress, starvation, and nutrient deprivation. And more importantly, this system is not unique to humans. It is found across all eukaryotic organisms, from simple yeast cells to complex human tissues. For many years, scientists knew about autophagy existed, but they did not fully understand how it worked at a genetic level. So that changed with the work of Japanese cell biologist Yoshinori Ohsumi Yoshinori Ohsumi in the 1990s. So Ohsumi, he used yeast cells as a model organism to study the autophagy process. By inducing starvation in these cells and observing them under the microscope, he identified key genes which were responsible for forming autophagosomes and regulating the process. These genes are now known as ATG genes. Okay, they are revealed in the molecular machinery that controls the autophagy. His discoveries showed how cells sense nutrient levels, activate autophagy, and break down cellular components for recycling. Because this work fundamentally transformed our understanding of cell biology, Yoshinori Ohsumi was awarded 2016 Nobel Prize in Physiology or Medicine. Today, his discoveries continue to influence research into aging, neurodegenerative diseases, infection, cancer, metabolic disorders, etc. Now, autophagy is not a random process. It follows a very organized four-step cleanup cycle, which is there inside the cell. The process begins with the start phase, where there's a tiny membrane structure called as a phagospore, okay, which begins to form. So think of it like a small bubble starting off to grow inside the cell. Next comes the wrapper phase. So here the membrane stretches and surrounds the damaged cellular components such as broken mitochondria or misfolded proteins. Eventually they seal them inside a closed structure called a autophagosome. Fine. So in the joint step, this autophagosome it travels through the cell and fuses with the lysozyme, which is filled with powerful digestive enzymes. Finally comes the breaking down and the reuse. Okay. So the cells break the captured material into small molecules like amino acids and fatty acids, which the cell can recycle for energy or to build up new cellular structures. In this way, autophagy acts like a cellular recycling center, keeping the cell clean and efficient. It's similar to how how a waste, like it's kind of similar to how wastes are disposed, waste disposal procedure, right? Kind of similar to that, analogy to that, right? Yeah. So, so this is how, now let's look at the molecular steps involved in autophagy. First one is initiation. First step is initiation. Here there is a membrane structure called as the phagophore which forms. It begins to form in response to signals like nutrient deprivation. In the step 2, right, elongation and capture, the membrane expands and wraps around the damaged proteins, organelles or cellular debris. Third step is fusion. Here it fuses with the lysozyme. The completed autophagosome, which is there, it creates a structure sometimes called as autolysozyme. Finally, the fourth step, degradation and recycling. So the enzymes inside the lysozyme, they break down the contents into small molecules, and these recycled small molecules, they are then released back into the cytoplasm where the cell can use them for energy production and cellular repair. This efficient recycling system helps cells to survive periods of stress, starvation, and nutrient scarcity. Cells constantly monitor whether nutrients are abundant or they are scarce. Two key molecular sensors help control this balance. One is mTOR and the other one is AMK. When the nutrients like glucose and amino acids are plentiful, the protein complex, which is mTORC1, right? mTORC1 becomes active. You get mTOR. So mTOR, it acts like a growth signal telling the cell that the resources are available. So autophagy is turned off. But while fasting or during energy shortage, during nutrient deprivation stage, the situation changes. As cellular energy drops, the ratio of AMP, okay, to ATP increases, activating a energy sensor called the AMK. So the AMK then switches the cell into a conservation and repair mode. It inhibits mTOR and activates proteins like ULK1, these kind of proteins, ULK1 ATG proteins are activated, okay, which begins to build the autophagosome membrane. This molecular switch allows the cell to quickly adapt between the feeding and fasting states. It ensures that the survival during times of nutrient deprivation or nutrient stress. Fine. So here, see, these are the key molecular regulators. First is mTORC1. This is a nutrient sensor, so it stops the autophagy when nutrients are abundant. You have AMK, which is an energy sensor, so it will activate autophagy when energy is less or energy starvation is there. Okay. Then you have proteins, ULK1 and ATG proteins. These are executioners, you can say. They build up the autophagosome membrane. So these switches allows the cells to respond quickly to the feeding versus the fasting time, fasting states. Okay. So how exactly does fasting activate autophagy inside our cells? So it starts with a change happening in the nutrient signals. So when we eat, hormones like insulin and IGF-1 increase and amino acids become abundant in the bloodstream. These signals activate a protein complex called mTOR, which tells the cell that nutrients are available and growth can continue. In this state, autophagy is mostly suppressed. But during fasting, nutrient levels starts to fall. So that time, the lower insulin signaling and reduced amino acid availability cause mTOR activity to decrease. At the same time, the cell senses an energy shortage. As ATP levels drop and AMP levels rise, an energy sensor called AMK becomes activated. AMK then phosphorylates proteins like ULK1, which initiate the autophagy pathway. So when mTOR inhibits the AMK and when mTOR is inhibited and AMK is activated, the brakes on the cell's recycling system are removed and the autophagy begins. This mechanism has been clearly demonstrated in many cellular and animal studies helping to explain how fasting and can trigger cellular cleanup. So once the fasting signal is detected, the cell must convert the signal into action. This is where ULK1 complex and ATG proteins come into play. The process starts with ULK1 complex, which acts like a command center for autophagy initiation. So when the AMK activates ULK1, it sends a signal to a group of genes called ATG genes, okay, many of which were discovered through the work of Yoshinori Ohsumi. These ATG proteins, they function like the execution machinery of the autophagy. Okay, they are the execution machinery, executors of autophagy. So they assemble and expand the autophagosome membrane, which is there, and around the damaged cellular components. Step by step, these proteins physically they build the structure which captures the cellular waste and it delivers it to the lysozymes for degradation. In other words, you can say that the signal from fasting is translated into a coordinated molecular construction project, resulting in the formation of autophagosomes, which begins the cleanup process. Okay, you can see here. So, yeah, fine. So, this is how it works. Now, many discussions about fasting and autophagy often mention a 16-hour threshold. But what does this actually mean biologically, right? So early in the fasting period, the body still relies on recently consumed nutrients and stored glycogen for energy. During this time, insulin and amino acid signals remain relatively high, keeping the mTOR active and autophagy relatively low. As the fasting continues, glycogen stores gradually decline and the body starts to shift its metabolism. Around 12 to 16-hour mark, nutrient signals decrease significantly, while as the cellular energy stress increases. So this activates AMK and gradually suppresses mTOR activity. As a result, autophagy activity begins to rise, entering what we call as a cellular cleanup phase. However, it's important to understand that this is not an exact switch that flips at precisely 16 hours. It is more of a gradual metabolic transition. Fine. It's not like a switch. It happens gradually. So what does scientific evidence actually tell us about fasting and autophagy? In animal studies and cellular experiments, nutrient deprivation reliably activates autophagy. When cells are deprived of nutrients, researchers consistently observe increase in the autophagosome formation and recycling activity. In humans, however, the evidence is more complex. Directly measuring autophagy inside human tissues is technically difficult, so studies rely on indirect markers like changes happening in gene expression, metabolic pathways, or protein levels. Research on intermittent fasting has shown shifts in these biomarkers, which suggests that increased autophagy related activity. However, controlled human studies proving a precise rule like 16 hours of fasting equals autophagy activation are still limited. So while fasting clearly influences cellular metabolism and likely promotes autophagy, the exact timing and the magnitude can vary between individuals. So why is autophagy so important for our health? One major benefit is quality control inside cells. By removing the damaged proteins and worn out organelles, autophagy helps to maintain the efficient cellular function. Autophagy also plays a role in protein management. It prevents the buildup of harmful protein aggregates which can interfere with their normal cellular activity. Another important aspect is disease, its role in disease relevance. Autophagy helps to remove intracellular pathogens and damaged cellular components, which contributes to infection control and overall cellular defense. Finally, autophagy supports metabolic flexibility. By recycling cellular components and releasing basic molecules, it helps the body to adapt to changing energy conditions like switching between feeding and fasting states. In many ways, autophagy acts like a cellular maintenance system, keeping cells clean, efficient, and resilient. The effects of autophagy extend far beyond simple cellular cleanup. In neurodegenerative diseases like Alzheimer's and Parkinson's, the buildup of misfolded proteins damages neurons. Autophagy helps to protect these cells by clearing away these harmful protein aggregates. Autophagy also plays a role in cancer biology. In early stages, it can suppress tumor formation by removing damaged cellular components. But however, in established tumors, cancer cells can sometimes use autophagy to survive stressful conditions. Another important aspect is infection control. Okay, so autophagy can directly capture and they can destroy the intracellular pathogens through a process called xenophagy. Finally, autophagy is strongly related to aging and longevity. So by repairing the cellular damage and maintaining protein quality, this process can contribute to healthier aging and longer lifespan. This is why autophagy has become a major focus of research in modern medicine. Fine. So how can we apply this scientific understanding to everyday life? So one approach that has gained attention is time-restricted eating, a form of intermittent fasting. So how does it work? So the idea is to maintain low nutrient signals like reduced insulin and mTOR activity for long enough to activate cellular repair pathways like autophagy. So a common method involves finishing dinner in the evening and delaying the first meal until around the midday next day. It creates a fasting window of roughly 16 hours. During this fasting period, hydration remains important, so water, unsweetened tea, or black coffee are generally allowed since they do not significantly stimulate nutrient signaling. While this approach is widely studied, it's important to remember that individual responses can vary, and fasting practices should always be approached sensibly and safely. Fine. As we conclude, it is important to keep a balanced perspective on fasting and autophagy. First thing is the individual variation matters. So the timing and strength of autophagy activation can differ depending on factors like age, genetics, the metabolic health, and lifestyle. Second, autophagy is context dependent. So while it can support cellular repair and stress adaptation, excessive or inappropriate activation can not always be beneficial, especially in certain medical conditions. And third, fasting should never be viewed as a replacement for proper medical care. While it may support metabolic health and cellular maintenance, it is only one part of a much larger picture that includes nutrition, sleep, exercise, and medical guidance. Understanding the biology helps us to appreciate how our bodies maintain themselves, but science also reminds us to apply these insights thoughtfully. So thank you everybody. I'll be giving the links for these link of all the references and materials in the description box. You can check that out. Fine. Thank you everybody.